to Dr. Michael Levy, who's going to talk about research in TM.
So if you stop eating, then your brain is going to be susceptible to all of the base in your blood, and you'll fall asleep because all of the acid is being used to digest your lunch. But if you continue eating, your brain will stimulate your brains you'll be stimulated enough to go through. I think we have 2 more presentations, and mine will be pretty quick. My focus is going to be on research in transverse myelitis, and I'd like to take you through 3 studies, just introduce them to you so you know the kind of work that we do. And if you think you may be eligible to participate, then that would be great. We have a study coordinator outside who would love to sign you up for these. And if you're not eligible, then tell your friends, tell other people who might be interested. I'm going to focus on 2 different types of transverse myelitis, or 2 different phases. The first is the acute phase. So this is when you come in and you're sick. You first have your symptoms of weakness, numbness, bowel bladder problems, whatever it is, and you come into the hospital. Most of the time nobody knows what's going on, and this is your acute illness. This is acute transverse myelitis. And we have a trial going on in a specific population of transverse myelitis where we try to limit the damage, and that study is called the SynRISE study. I'll talk about that. Then for everyone here, I assume, who's not in the acute phase, who's now recovered partially or maybe not made any recovery, and who still have damage and need to recover from this damage. And you could be managed for spasticity and depression and all of the other problems, but if you really want to try to cure or really overcome your disabilities, then the focus of our research is to do that. And we have 2 areas of active research. One is in stem cell research to actually regenerate the lost spinal cord tissue. And the other is the Ampera trial that Dr. Zakowski mentioned, which while we're not regenerating tissue, we're trying to make it work better. So let me show you a video of what the complement system does. The complement system is a part of your immune system that does the damage. And let me see if this focus in on this. So when you get bacteria into your blood, you have proteins that attack it. And the ones that do the most damage are a group of proteins called the complement proteins. And I mention this because it's important in TM. This is an example of bacteria. And you're going to see the complement proteins here floating around. They don't really do any of the thinking. They just attack. Okay? And as soon as they bind onto this bacterium, they form a complex with other complement proteins, and it sort of causes a cascade effect. You're going to see this bacteria explode. This is great for bacteria, okay? So if you get infected with bacteria, you want a complement system to destroy it. The problem is, if you don't have bacteria and you have your complement system activated in your spinal cord of all places, then that's not so good. Turns out that's what happens in transverse myelitis. So this is a slice of a spinal cord up here. This is a slice taken out where you can see a whole quarter of it is damaged, and this is from transverse myelitis. And what we've noticed about a decade ago now is that complement, which is stained here in red in the tissue, is activated in transverse myelitis and affects a lot of the damage. So the immune system attacks the spinal cord and triggers the swelling and the inflammation, but what actually causes the damage is that complement system, which is just reacting to the environment, but what it's doing is it's essentially blowing up your spinal cord. So the thinking here is, can we limit the amount of damage? Not by modulating the immune system, which is what you currently do. When you come into the hospital, you get your steroids, you get plasma exchange, you get whatever you need to do to calm your immune system down. But can we also target the complement system to mitigate the damage? So there are 2 drug companies now that have approved medications in the U.S. that target the complement system, and these are new. And one of them is called SynRISE. And Cinryze is an intravenous medication that's approved for a different indication, but they approached us because we have a large transverse myelitis center, and they said, hey, this is a great idea. Let's work together. We'll give you the drug. You give us the patients. So patients are admitted here to Johns Hopkins with a certain type of transverse myelitis. We give them the complement drug, and we see if we can reduce the damage. So here's the idea. You come in with your acute TM. You still get the steroids, as you normally would, and if you need plasma exchange, you would get that as well. But in addition, you get the Cinryze drug. We've now recruited 9 patients out of 10. This is just an initial study to see if it's safe and to see if there's any promise to move forward. And while I can't release any of that data, I'm under contractual obligation to remain silent. It does seem to be helpful in some way, and I think we'll move forward with a larger trial where we'll have a placebo group. Again, this is designed for acute transverse myelitis. So anybody here who has a recurrent disease, for example, this might be helpful for you in the future if you have another transverse myelitis, or if you have patients or neighbors or anybody else out there who has an acute transverse myelitis. You may want to alert them, hey, there may be a drug out there that could reduce the damage from your myelitis. It's given in addition to standard of care, and it turns out to have an excellent safety profile. So that's for the acute phase. And what about for the phase of transverse myelitis long after the damage was done and there's ongoing disability? How are we going to improve their function? So let me talk a little bit about our stem cell research, because this is really, for long-term promise, this is really where the greatest research interest lies. And what are stem cells? Stem cells are simply cells that have the capacity to proliferate and become other cells that you direct them to become. In the case of transverse myelitis, what you want are to replace 3 cell types: the neurons, the supportive cells, the astrocytes, and the myelin-producing cells, the oligodendrocytes. All 3 of these cells can be produced by stem cells that come from a variety of sources. The politically charged cells are the embryonic stem cells because they come from embryos. But the new breakthrough stem cells that come from adults, for which the Nobel Prize was just awarded, are now being used a lot more than embryonic stem cells. I don't think there are any more clinical trials with embryonic stem cells, because now we can take your skin cells, for example, culture them in a dish, take them back into stem cell form, and then move them forward into neurons, astrocytes, and oligos, and then transplant them right back into you. So that's kind of the new phase of stem cell research that doesn't have any political implications. So we can take these stem cells and put them into mouse models, which is all we have approval to do right now, and show that they can make myelin. 2 companies now have FDA-approved cells that are in experimental trials in humans. They're not in transverse myelitis trials, they're for trials for other diseases, but they sent us their cells, and they said, can you show that these cells have the capacity or the potential to make myelin in this mouse model that doesn't have any of its own myelin? So no, I'm not sure how well you can see this red, but this red highlights protein, myelin protein, made by stem cells that are in green. Again, I'm not sure how well you can see that, but we transplanted these stem cells into mice that didn't have their own myelin, and the stem cells migrated from the back of the brain all the way forward to the optic nerves and all the way down the spinal cord, and they produced myelin because this mouse in particular needed it. So they're responsive to their environment. This is a stem cell, like I said, it's being used for other clinical trials, particular ALS or Lou Gehrig's disease. But the company figured, well, if it can make myelin, well, then maybe it would be useful for transverse myelitis. So we contracted with them to develop this preclinical model to show the FDA that this is possible and safe in humans. And what we have to do is show them that these stem cells have the potential to produce myelin, like I just showed you. We have to show them that they migrate. So, in transverse myelitis, it's very convenient because there's only one lesion. You can inject the stem cells right into that lesion. That's why it's a great proof of concept model. But for diseases like MS and other diseases where there are lots of lesions, it's too difficult to make all these injections. And so you want to be able to show that the stem cells can migrate and can go only to the places where they're needed and not to the places where you don't want them to go. You also want to ensure that these cells survive. You don't want to go through this stem cell injection only to have them killed off by your own immune system again. So we have medications now that can suppress the immune system in the correct way so that these stem cells survive. And then the hardest part actually is to show that these stem cells can integrate. You can imagine the spinal cord is very complicated, as is the brain. And if you're going to put in these cells that have to regenerate the previous tissue that was there, they have to connect with the brain cells that are sending the signals down. They have to connect with the muscles and the nerves and the rest of the body, and that can be very difficult. Now, while these cells have a lot of potential, there's still a lot of training we have to do to get them to restore function. The company that we're working with has filed an application with the FDA to move forward with a trial in transverse myelitis. It's in collaboration with two groups. The first is the company, NeuralStem. They're based in Rockville, Maryland, and our second collaborator is the NIH. They have a new center there called the Center for Regenerative Medicine. They have a mandate to move forward with clinical trials. Stem cell biology, there's only one person working there right now, but this is an institute at the NIH that just started, and they are poised to move forward with several clinical trials. It will likely be based in Bethesda. There is an NIH hospital that's considerably cheaper than ours, so if you get a surgery here that costs $100,000, you can get it done at the NIH subsidized for just $30,000. So that's very enticing. And they're just down the street, about 45 minutes south, and so that's not too difficult for many of our TM patients, hopefully. We'll try to recruit 6 patients to this initial study. We want to get 3 who have absolutely no motor, sensory, or bowel or bladder function below the lesion. And for this, we want to demonstrate to the FDA that these cells don't cause any more harm. You can say, well, how could it cause any more harm? I don't have any motor function. I don't have any sensory function or bowel bladder function. How could it cause any more harm? Well, it could form a tumor, or the stem cells could migrate to the wrong place. It can get into the brain or somewhere else you don't want it to go. So really, these first 3 patients are just to demonstrate we're not going to cause any more harm. Then we're going to wait 6 months, and then we're going to try to recruit the next 3 patients, which are what we call ASIA-C. This is a scale that demonstrates you have some weakness, some sensory loss, and some bowel bladder dysfunction, but not complete. Because what we want to show here is that adding the stem cells could actually improve the function. And then once we demonstrate that, we can move forward with phase 2 studies. These stem cells will be directly transplanted into the spinal cord lesion. The company has a big setup that they train their neurosurgeons to use, and you'd be in the operating room, and they would attach this setup to you so that when the needle is lowered into your spinal cord and you continue breathing, the needle moves with your body, and so it doesn't cause any more damage. It's very invasive. It's a surgery. It takes some time to recover from it, but we are hoping that this really can show that stem cells have the potential to actually regenerate spinal cord tissue. Okay, Dr. Zakowski mentioned this trial. This is the Empira study. This is a lot less invasive than a stem cell transplant. And you already heard a lot about how this is designed. Let me just mention the highlights. So in TM, there's just one lesion. And for Empira, which is already approved for MS, MS patients have a lot of lesions, and a lot of lesions in the same pathway. And if Empira can help those patients walk, the thinking is that with just one lesion to overcome, Empira has a much better chance. Actually, got to have lunch with the CEO of the company that makes Accorda, and I said, why didn't you use TM patients for your first trial? It seems like, an easier option, much more chance of success. And he said, well, that was my first option. He's a neurologist. He said transverse myelitis is the best proof of concept model we have for this, but the business office said there's no money to be made. You guys, there aren't enough TM patients out there, so we moved forward with MS, and now they're making lots of money. But they are very interested in TM, and the CEO himself is interested in TM, and one of his goals in life is to create drugs and devices that can help people get out of wheelchairs. That's his personal goal. So he liked our idea to test this drug out in transverse myelitis personally, and is very interested in the results. And as Dr. Zakowski showed you, when you lose myelin on your axons, then the electrical signal, which is mediated by ions, by salts, is dissipated out. Those ions are potassium in this case, and the drug just blocks the potassium channels. So the ions can't get out, and they continue to diffuse down the axon and hopefully continue the signal down the axon. It's a very simple idea, and the drug's been out for something like 40 years. It's been available for a long, long time. But the reason that this drug company has the patent is because they made it safe. So there's much less risk for seizure. And I just want to show you a video of a patient on the drug 3 weeks after starting. Let's see if this works. Let's see, I'm not sure if it's working. Basically shows a woman walking down a hall. This is our standard time 25-foot walk. What she does is she's taking Ampera for about 3 weeks. This is her pre-trial walk, and this is after taking Ampera. And we're not talking about miracles here, okay? We're not saying that she's going to, throw her cane down and start doing a dance, but we are saying, look at how much faster she's walking. On average, MS patients walk 20% faster. We're thinking TM patients can do even better. This is her going back. So you can see she has the same disabilities, the weakness is in the same place, but kind of all the muscles seem to be working a little faster. Gait's a very complicated system, but her walking improves on the drug. This is the design. Everybody gets a chance to take the drug. You get randomized into one of two arms when you come visit, and you either start on the placebo or the drug. You don't know. You may have side effects, or you may think you have side effects. You may think you walk better, and you can come to me and you go, I know what I'm on. They often say, I know what I'm on. I'll say, I don't even know what you're on. Nobody knows except the pharmacist. So you don't really know. But then you get switched. When you come back, you get switched to the other arm. So you are your own internal control. So I'm not going to say you're walking better because compared to 10 other patients, you're walking better than them. I could say you're walking better because compared to you, you're walking better. Or I may say, this drug is really not that helpful for you because, look, when you're on the drug and when you're on placebo, you have the same walking speed. But this is the idea behind the trial, and if it's successful and if it's helpful in TM, then it would be available for other TM patients in the United States. It would not be FDA approved, but it would be covered by insurance companies. We currently have 7 enrolled. We're looking for 18 more patients. If any of you want to participate, Wah is sitting outside at the table and can give you some information. The big catch with this trial is you have to visit us every 2 weeks. So for those of you who live in Kentucky or other places that are far away, we do have one participant from Georgia who comes up every 2 weeks. So it's not that hard, but we do need to see you every 2 weeks for the trial. And lastly, I just want to mention a few futuristic ideas. Transverse myelitis is a very interesting scientific problem for the recovery folks because there's just one lesion. It's very rare in the nervous system to have just one lesion that you need to overcome, for which we understand a lot of the pathology. So there are already a group out there, one from the Mayo Clinic, that's moving forward with the clinical trial to use a product called R-IgM22. This is an antibody. It was made by a human being who had an amazing remyelination potential. They isolated it from her and then made a synthetic form of it. And they infuse it. Now they're launching into multiple sclerosis, of course, because that's where the money is. But the same idea is true in transverse myelitis, that after your acute phase, about 2 to 4 weeks after your initial transverse myelitis, if you can get this drug, there's a good chance that you will remyelinate. And so there's a very strict time window, and we're not 100% sure where that window closes after your myelitis, but this is something that is moving forward. Then Dr. Becker mentioned that you have a lot of stem cells of your own already. You don't need me to surgically implant them. You already have a lot of your own. The problem is they're not doing what we want them to do or what we hope they're doing. They just kind of sit around. It's really remarkable. If you track these cells after a transverse myelitis event, they do migrate to the lesion. And you think, wow, they're really responding. They're about to form new myelin. But then they just stop. They just stop in their tracks. There's some sort of inhibitory signal there that we've evolved over time to just tell these stem cells, don't do anything, just don't make it any worse, potentially. And so those stem cells just sit around and don't do anything. If we could harvest the potential of your own stem cells to regenerate your spinal cord tissue, that sounds like the best of all situations, and we have an active group at Hopkins doing that. And then I did mention for those where we can't stimulate the endogenous stem cells, then we are going forward with transplanting other stem cells that we can genetically modify to be more resistant to the environment, so that even if there is an inflamed environment, they can survive and regenerate tissue. So I will stop there. I'm at my 20-minute mark. I'll take a few questions. Mm-hmm. Yeah, we're hoping. Yeah, the NIH has to approve, the FDA has to approve it. We're expecting that to take about 3 or 4 months, and then we're going to put all the funding together. And there are several groups that are interested in doing that. I would say hopefully by next year, next summer, we'll get this all together.
We are recording this, so if you wouldn't mind waiting for the microphone, that would be awesome. Anyone else?
Is Ampera the same as AP4?
Yeah, 4AP is the old drug. It's been around for a long, long time, and Ampera is just a formulated form of 4AP that it's bound to a resin, so it's released in a much slower and controlled way, so that it doesn't cause any of the same side effects that 4AP does, or fewer.
Is the effect any better than the original compound?
It turns out it is, because in order to get the best effect from 4AP, you may have to give a really high dose, and that would cause a seizure. But with Ampera, you can give it in twice a day, and eventually the dose reaches that sort of optimal range, so you do tend to see better results with Ampera.
I'm not sure if I understand. If your own stem cells won't prevent that, if you inject other stem cells, we know it will help, or is that the purpose?
Well, the stem cells that we're going to put in, we're hoping are more resistant to the inflammation that caused your transverse myelitis and that caused your endogenous stem cells not to activate. So these are very different cells, not like the ones in your own body, but ones that we hope are more resistant. Yeah.
Yes, if I understand correctly, the 4AP, if it works successfully, it's going to repair all the myelin, and hopefully improve patient's gait. But what about the actual nerve cells that are inside or within boundaries of myelin where there's been damage? It's not going to do anything for them, is it?
So Ampera actually works on the nerve cells themselves. They don't affect its health, ultimately. What they do is they just close their potassium channels so the signal gets through better. It doesn't keep the neuron alive any better, and it doesn't stimulate any myelin production. All it does is it just kind of turns up the volume a little bit on the neurons, and that's all we're asking it to do. So if you don't take the drug, then your function is a little worse, and if you do take the drug, function a little better, but there's no sort of long-lasting effect.
Okay, last question, and then I'm going to pass out some more cards during the break. So if you have any to write down.
So if you do the transplant and inject it to the one lesion on a TM patient, and the stem cell goes to somewhere else, what potentially could happen there?
Worst case scenario? Worst case scenario, it forms a tumor. That's kind of what everybody's big fear is. It's going to get into the central nervous system. It has the capacity to proliferate. It's going to lose all control, and it's going to form a big tumor, and you're going to have brain cancer. That's the big scare. It turns out it hasn't done that yet. There have been now 15 patients with Lou Gehrig's disease transplanted, no tumors. The first embryonic stem cell transplantation by Geron in 2010. 2011, 5 patients transplanted in the spinal cord, no tumors. So we now have experience from mice. You can do it in certain ways that they'll form tumors, but the usual way they don't form tumors either. Yeah. Thank you. Now up